Ladies and gentlemen, I am Alyssa.
I'm Shirmei.
I'm Charlene.
I'm Yun.
And we are the innovators of this
ACNCE Insecticidal Repellent.
And we are honored to represent SMJK Heng
In this competition.
Now, before we dive into our presentations, I would like to ask you, have you ever wondered the food price is getting skyrocketing lately? Ever wonder why? The answer actually lies in this food insecurity that is driven by climate change and also this one unexpected invader, the pest. Among all of the pests, stand out this one silent killer, which is the fall armyworm. In just a few decades, this alien species has been attacking over 350 different species of plantations, devastating maize crops, and slashing up to 100% of yield productions in Africa. Now, shifting our attentions to the queen of cereals, which is also to be referring to be the corn in comment, it is shown by the study that up to 20 to 50% on the global scale of crop loss that is damaged due to this fall armyworm attack. Shocking, isn't it? But now that we know the enemy, the real question is that how are we going to fight back? Well, let's find out. Before we present you our strategy, please allow me to share you a little information about our enemy, shall we? The fall armyworm has a simple yet destructive life cycle, which is first hatching from the egg, growing into a larvae, and lastly emerging as a moth. The fall armyworm larvae is divided into 6 instarts, which is for the first 3 is called as a young larva stage, and the later 3 is categorized as the elder larva stage. The larva of the fall armyworm is just like a teenager growth spurt. They eat non-stop. As to counter this, we have designed this ACNCE Insecticidal Repellent, which is in the full form of the full name. We call it as allicin, capsaicin, and neem combined extract insecticidal repellent as to control and to repel the fall armyworm. With this approach, we believe that it will maximize the effectiveness in protecting yield productions from the fall armyworm attack by targeting its ability for its strong adaptations, high-speed migrations and spreading, rapid reproductions, and most importantly, high effective digestion. With this, I am confident it has the push and proof of the potentials of our project to taking down the bigger struggles in the agriculture.
Our solution combines 3 powerful natural compounds. Firstly, allicin, which is extracted from garlic. It is a natural antimicrobial agent that disrupts the metabolism and cell structure throughout its gut. When fall armyworms encounter allicin, it disrupts their feeding behavior and deters them from consuming treated plants. Secondly, capsaicin, that is extracted from chili. It is known for its insect-repelling properties and irritating the fall armyworm's nervous system. Capsaicin has a chemical structure that allows it to interact with the nervous system of the fall armyworm. Its structure includes a vanilloamide group, which is responsible for pungent properties and its ability to bind to sensory receptors in fall armyworm. When capsaicin binds to these receptors, it disrupts the normal function of fall armyworm's nervous system, leading to symptoms such as hyperactivity, erratic movement, and eventually paralysis. This disruption affects the insect's ability to feed, grow, and reproduce, making capsaicin an effective natural insecticide. Thirdly, azadirachtin, that is extracted from neem. It is a well-known botanical pesticide that has potential to destroy its reproductive system, thus controlling the population of fall armyworm. Chemical structure of azadirachtin is similar to insect hormones called ecdysones, which controls the process of metamorphosis in insects. It affects the corpus caardiacum, which controls hormone secretion. Azadirachtin appears to function as the ecdysone blocker, inhibiting the production and release of these vital hormones. The prothoracic glands of the fall armyworm produce ecdysone, a hormone that triggers metamorphosis and stops growth in the larva. One of the PG-enriched genes, the nuclear hormone receptor 3 was decreased after azoxystrobin treatment, proving its effectiveness. Why do we choose these 3 compounds instead of others? Because together they form the ultimate defense, that is, cost-efficient, highly effective, sustainable, and eco-friendly. This trial targets the very traits of 4-armyworm that has strong adaptability towards chemical pesticides, rapid reproduction, and high migration speed. With this innovation, we are not just fighting for armyworms, we are changing the game.
Well, we test each formulation on 4 animal specimens, measuring the repellency rate by recording the time taken it took for larvae to leave the treated constants. The most effective concentration was stabilized with glucopure to enhance the stability of oil in water. Furthermore, the GCMS analysis confirmed the presence of key bioactive compound, ensuring the potency of AC/NCE insecticidal repellent as the natural insecticidal repellent. Moreover, we determined the repellency by assay through AC/NCE insecticidal repellent by calculating the repellency rate. If the result was exceeded the standard value, it was classified as repellable. If it was less than the standard value, it was considered as non-repellable. In the last experiment, we determined the pH value of soil to identify the impact of AC/NCE insecticidal repellent towards the pH value. There are 3 types of soil we test, which is chernosem, pit soil, and sandy loam. These 3 types of soil we test with distilled water and AC/NCE insecticidal repellent separately. At last, the result shows that our AC/NCE insecticidal repellent slightly neutralized the pH value of soil. This indicates that our AC/NCE insecticidal repellent is environmental friendly.
Moving on to the result and analysis. The formulation was developed using 0.3% of allicin, 1.2% of capsaicin, and 1.2% of neem oil, as this concentration demonstrates the optimum performance in the previous test. One-way ANOVA analysis shows that there is a high significant difference between the group means, as the p-value is lesser than 0.05, which is lesser than 0.001. A larger f-value combined with a very small p-value means the variation between the groups is much greater than the variation within the groups. Post-hoc test as well reveals that there is a significant difference between the AC/NCE group with allicin group, capsaicin group, and neem group, as the p-value are all lesser than 0.05, which is lesser than 0.001. The repellency bioassay result shows that the concentration of allicin, capsaicin, and neem oil from 0.3% to 1.2% are all effective insecticidal repellent. The pH value of different types of soil, which is the chernosem, pit soil, and sandy loam, has all increased after interacting with our AC/NCE insecticidal repellent. This indicates that our AC/NCE insecticidal repellent is environmentally friendly, as it neither acidifies nor alkalizes the soil. In conclusion, it is valuable to utilize the AC/NCE insecticidal repellent with 1.2% of concentration as a sustainable and environmentally friendly insecticidal repellent against 4-armyworm in the maize field. Thank you.